AS level biology 9700 topic 1 cell structure. Students, already I have discussed part 1 and part 2 of this topic cell structure. Today we are going to discuss part 3, and in this part 3 we will discuss the ultrastructure of endoplasmic reticulum, because previously in the second video I had discussed the ultrastructure of an animal and plant cell. Now the different organelles of the cell will be discussed in this part. So first of all, the ultrastructure of endoplasmic reticulum. And endoplasmic reticulum, uh, they are these cell organelles. They are present in both animals and plant cells. And endoplasmic means within the plasm, that is, they are in the cytoplasm and in the form of a network, a tubular network. So, tubular network or flat vesicular network. So, their structure is just like tubes and flat vesicles, and they are present in the cytoplasm, and the tubules and vesicles interconnected with one another. That is, they are interconnected with one another because they have to perform the process of transportation or the delivery system of the cell. So they are interconnected with one another to transport the different, uh, you can say, the substance which are formed inside, as for example, the lipids and protein molecules. They should be transported over there. So the transportation will take place through these endoplasmic reticulums. So we can say this, it is an internal delivery system of the cell. So it make up approximately 12% of the cell volume. 12% of the cell volume. So it make the uh, highest percentage, as you can say, out of one cell which is already very smaller in size, cannot be seen with the eye. Without microscope it can be observed, so or cannot be observed. So 12% of their cell volume will be this endoplasmic reticulum, because in this way the proteins which are needed uh, to the cell, within the cell or out of the cell, they should reach to all parts inside the cell. So here you can see the diagram of endoplasmic reticulums, and you can see the both types of endoplasmic reticulum has been shown over there in this diagram. The nuclear envelope, they are originating from the nuclear envelope, and then you can see rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum because the ribosomes are attached on them. Due to the presence of these ribosomes, they look like the rough structure. So this rough structure is due to the presence of ribosomes, because the ribosomes are involved in the protein synthesis. So the smooth endoplasmic reticulums, they are without ribosomes. So both of they perform the different functions. So we will move towards the next slide, and in next slides we will be able to differentiate between these two types of endoplasmic reticulums. As already told you that the rough endoplasmic reticulum is has the ribosomes dashed on it. So these studied with ribosomes on cytoplasmic side protein. The function of rough endoplasmic reticulum is to help in the protein synthesis, because the function of the ribosomes is protein synthesis. So endoplasmic reticulums are involved in the protein synthesis. So they do not only the synthesize um, protein synthesis, but also modifies the protein which is formed over here. How do this modification takes place? The modification by adding sugars into it, that is, the protein is changed into glycoproteins. So rough endoplasmic reticulum not only involved in the protein synthesis, processing of proteins, but also modification of the proteins. And during this modification, they add the sugar into it, and then the protein is changed into glycoprotein. Now the other type of endoplasmic reticulum which don't have ribosomes attached on them, so they look like very smooth structure. So they are not involved in the protein synthesis. Mostly their function is the synthesis of lipids. Synthesis of lipids. So the site of various synthetic processes, detoxification, storage, and forms transport vesicles. Transport vesicles, you can say the Golgi bodies. So these transport vesicles are also formed by the smooth endoplasmic reticulums because they don't have ribosomes attached on them. So they are involved in the synthesis of lipids. So it was the function of both types of endoplasmic reticulum, which is a very precise transporting system or delivery system inside the cell. Now the next organelle, very important organelle, which is involved in protein synthesis. So the ribosomes are said that make proteins. So they make proteins, and why protein formation takes place in the cell? Why cell needed that protein? Because the protein is needed for many cellular functions such as repairing of the damaged tissues, or repairing of the damaged cells, repairing of the, you can say, lost or the damaged parts, so directing chemical processes as well. So ribosomes can be found floating within the cytoplasm. They can be freely present in the cytoplasm and also on the rough endoplasmic reticulum. That is two locations. Freely floating, they are freely floating in the cytoplasm. And secondly, they are present on the endoplasmic reticulum. So these are the two locations of these ribosomes. They are involved in protein synthesis, very important function. So ribosomes are made up of proteins and ribonucleic acid. Ribonucleic acid or RNA, so in almost equal amount. So it comprises of two sections known as subunits. A larger, I would like to show the picture of these ribosomes in the next slide. You can see over here the two units of the ribosomes, and these two units, large units and small subunit. So I would like to add that the ribosomes are of two types. Types of ribosomes. They are 70S and 80S. So what is S stands for? The S stands for Swedberg unit. Swedberg unit. Which is the, you can say, which is the unit of measurement for the sedimentation coefficient. So it indicates how quickly a particle sediments in a centrifuge, which is related to both the size and shape of the particle. So here we will only discuss that these are the two types of the ribosomes, that is 70S and 80S, and S stands for Swedberg unit. So already told you, that is they are located at two places. So the, and the function of these ribosomes is the protein synthesis. So the location, so the different, they are when they are present on the endoplasmic reticulum, or when they are present in the cytoplasm, freely available in the cytoplasm, so they will make the different types of proteins at that time, because cell will determine that is which kind of protein it makes. So it will be the decision of the cell, because the need of the cell, according to which the ribosomes will make up the protein. So if the ribosomes are floating freely throughout the cell, it will make proteins that will be utilized within the cell itself, because they are moving throughout the cell. So the whole cell will be capable to get the type of protein which is needed by that cell. So it will be formed by those ribosomes which will be freely available inside the cytoplasm. So the cell will determine which type of protein is needed by the cell. So ribosomes, free and the bound ribosomes. Free means which are present in the cytoplasm, and bound ribosomes which are present on the rough endoplasmic reticulum. So are very much alike in structure. Their structure is same and are associated with protein synthesis and lipid formation, So around plus lipid formation. So around 37 to 62% of RNA is comprised of RNA and the rest is protein. And then, the prokaryotes and eukaryotes. Prokaryotes, which don't have membrane-bound nucleus or proper, you can say, nucleus. So prokaryotes have different types of ribosomes inside. So the prokaryotes have 70 S ribosomes. So 70 S ribosomes in which the little subunit will be of 30 S and the bigger subunit will be of 50 S. So 70 S ribosomes will be in prokaryotes and 30 S and 50 S. 30 S will be the smaller subunit and the 50 S will be the larger subunit in prokaryotes, whereas in eukaryotes, the proper cells which have proper nucleus, so they will have 80 S ribosomes. So the difference is there. That is, they will have the 80 S ribosomes. So 80 S ribosomes in which the 40S will be the smaller unit and 60S will be the larger subunit. So in this way, the ribosomes will be differently available in prokaryotes and eukaryotes. They will be different in size. They will be different in unit as well. So the subunit and larger unit will have their own sequence or their own components inside. So they will be different over there. Now we will move towards the next organelle, which is called as Golgi apparatus. You can see the Golgi apparatus on screen. Before going in this, I would like to discuss this slide. The Golgi apparatus, also called Golgi complex or Golgi body, is a membrane-bound organelle which is found in eukaryotic cells. Eukaryotic cells, and that is made up of a series of flattened stacked pouches called cisternae. It is located in the cytoplasm next to the endoplasmic reticulum, very close to endoplasmic reticulum, and near the cell nucleus. So while many type of cells contain only one or several Golgi apparatus, plant cells can contain hundreds. I will move back in the previous slide. You can see over here, the structure is very much alike with the endoplasmic reticulum. You can say the incoming transport vesicles. So these are the incoming transport vesicles which are coming from the endoplasmic reticulum. And you can see these pouch-like structure or sphere vesicle-like structure, which is cisternae. And see, these are the formation of secretory vesicles, or this is the, you can say, pinched off a vesicle, which is a new vesicle is pinched off from this cisternae. So in this way, a secretory vesicle will be formed having the secretory material inside. So I was discussing this part. So the Golgi apparatus. So it was just you can say stacked pouched cisternae. So Golgi apparatus is responsible for transporting. So this is the function. You can say it is function. It is structure. And one more thing I would like to discuss about about its structure, that is a little bit information I would like to give you. That is, this Golgi apparatus is formed from the small pieces of rough endoplasmic reticulum. I would like to write over here, formed from small pieces of rough endoplasmic reticulum. So these small form vesicles. So these small vesicles will join together to make the Golgi body. So chemicals which are made in the endoplasmic reticulum are collected into these Golgi bodies where they are modified. So it means the Golgi bodies also modify. We will discuss in the function. So small vesicles can then be pinched off of the Golgi body, carrying new chemicals away, which are secreted when the vesicles reach the cell membrane. So they are secreted from the cell membrane. So it means they are formed from the small pieces of rough endoplasmic reticulum, and then their function is this, that is the Golgi apparatus is responsible for transporting, modifying, and packing proteins. That is the protein which is formed in the endoplasmic reticulum. Then small pieces from endoplasmic reticulum are get separated and they join together to make the Golgi bodies. And those Golgi bodies then are responsible for transporting, modifying, and packing proteins inside and lipids into vesicles for delivery to targeted destinations. That is, in the form of vesicles, they will take them to the place where they have to be distributed. That is, they if they have to release out of the cell, so they will go very close to the cell membrane to go out from through the cell membrane by the process of exocytosis. So, or they will be used inside. So it depends. So as the secretory proteins move through the Golgi apparatus, a number of chemical modifications may transpire. Important among these is the modification of carbohydrate groups. Also with the Golgi secretory vesicles are proteases that cut many secretory proteins at specific amino acid positions. So it means there are also different types of enzymes beside. So they will help in the they will also help uh, in the formation of you can say the lysosomes. Lysosomes are also formed by these vesicles. So lysosomes, we will discuss in detail lysosomes in the next videos, inshallah. So lysosomes also contain the enzymes. And the next organelle, very important organelle of the cell, is the mitochondria. So mitochondria, you can see rod-like structure, and which consists of double membrane, outer membrane, and inner membrane. And you can see the outer membrane covers the organelle freely permeable. You can say this is the outer membrane which covers this whole rod-like structure. So the inner membrane, it has many folds inside. So these folds are called as cristis. So and in the you can say intermembrane space between outer and inner membrane contains cytoplasm. So and within the inner membrane, various protein and enzymes are there, and that part is called as matrix. So in this way, you can see its structure has double membrane. So, it is very important. Now, what is the function of mitochondria? So, function of mitochondria is, you can see, energy production. Energy production, mitochondria convert the energy, you can say the energy which is produced as a result of respiration, that is oxidative. Mitochondria convert food into energy through oxidative phosphorylation. Oxidative phosphorylation means ADP is changed into AMP is changed into ADP, ADP is changed into ATP. So phosphorylation is a process by which ATP formation takes place. So formation of energy takes place by this mitochondria. This is the first very important function, cell molecule production. So cell molecules are the fundamental building blocks of the cell. So they can be classified into four major classes as, for example, nucleic acid, proteins, carbohydrates, and lipids. So these are the formation, mitochondria perform the formation of these four different types of cell molecules, and then cell death. Very important. Mitochondria play a role in programmed cell death. Programmed cell death is also known as a cell demise or cell loss. We can say that when a biological cell ceases to function and dies, so it can be normal regulated process or a response to injury or stress due to any reason when the cell death. So its mitochondria also play a role over here. Then aging, mitochondria also regulate the aging process. So aging process, then ion homeostasis, mitochondria play a role in ion homeostasis. It maintains the balance of the different ions inside or outside the cell. So the calcium signaling is, you can say, signaling is a way of communication of cells with one another. So calcium signaling, it play a role in the signaling, that is calcium level is increasing or decreasing. So or calcium ions are moving out or of the cell or inside the cell, or they enter inside the cell. All it is regulated by the mitochondria. So these are the different functions of this mitochondria which are performed by these very special organelles. So these organelles also replicate by dividing into two, uh, using a process similar to the similar asexual form of cell division employed bacteria, which is called as binary fission. In which one uh, bacteria, or we are talking about mitochondria. One mitochondria is divided into two. So video microscopy shows that mitochondria are incredible dynamic. They are constantly dividing, fusing, and changing shape according to the need of the cell. They have to do that. They have to divide. So they have, if they don't need more energy, so they will fuse together. If the less amount of mitochondria are needed, so they will change the shape. They will fuse together. If the more mitochondria are required to produce more energy, so then they will divide. Their number will increase. So indeed, a single mitochondrion may contain multiple copies of its genome at any time. So they can make many copies. So logically, mitochondria multiply when the energy needs of a cell increases. They will multiply in number, they will divide, they will multiply, they will divide into 2, 2 into 4. So in this way, on this formula, the binary fission will take place, the division takes place, their number will increase. So power hungry, therefore power hungry cells have more mitochondria than cells with lower energy needs. You know, the different cells has to perform different functions. So according to their functions, they need more mitochondria or less mitochondria. So if the cells have more mitochondria than needed cell, then the mitochondria will divide immediately and their number will increase. Repeatedly stimulating a muscle cell with spur the production of more mitochondria. Repeatedly stimulating the muscle which is moving, its movement is fast. So at that time, mitochondria are increased. So because the muscle, when it is working, when it is contracting or relaxing, at that time, what will happen? The more energy is needed. So to compensate or to balance the energy needs of that tissue or the cell, the more mitochondria are needed. So at that time, mitochondria multiply and give their number is increased. So students, it was all about uh, part 3. Wait for part 4. But please don't forget to like, share and subscribe my channel. Thanks for watching.